Hi, my name is Haley Howard and I'm going to be discussing Dr. Beth Stevens' presentation titled Microglia Function and Dysfunction in Alzheimer's Disease. Now, microglia are very small cells. They're immune cells and they make up about 5 to 7% of brain cells. And they're interesting in part because they're the only cells in the brain which are not derived from the brain, they derived from the yolk sac. And their functions are incredibly dependent on disease context, injury context, and their physical location in the brain environment. Now, Dr. Stevens is an expert on this kind of cells, and her lab studies how the cells function in development when it's normal, healthy brain, and when there is a disease or some kind of pathology happening which causes the cell function to go awry. Now, Dr. Stevens in her lab, their primary focus is determining microglia functioning in the context of disease and injury, and primarily in the context of Alzheimer's disease. And so even though Dr. Stevens mentions a lot of past research, she does so to build a baseline and explain how we know what we know about microglia and where it came from. But the main focus of her presentation is the last research endeavor she mentions, which is the one which is currently going on in her laboratory. And that research study is going to find specific genes in pathways that are high risk for Alzheimer's that are also involved in microglia functioning. And then they're going to perturb those pathways or perturb those high risk genes and see what kind of functionality changes happen in the microglia from those pathways. So they'll be able to see, oh, if we mess with this one gene, the risk for Alzheimer's actually goes down, or if we mess with this other gene, the risk goes up exponentially. And so their hope is by just totally messing with all of the high risk genes, they'll be able to determine a very good explanation for how all of these separate pathways work. And that way they'll be able to vastly expand our disease pathology knowledge of Alzheimer's and eventually be able to say, okay, well, now that we know how the pathways work that cause Alzheimer's, we can work backwards and we can block those pathways. And their hope is to eventually use the knowledge of microglia to really decrease and possibly even end Alzheimer's disease. Now, this current study going on in Dr. Stevens' lab is, of course, specified to Alzheimer's disease, and they were able to determine that microglia have an impact in Alzheimer's disease by really getting a better idea, and this is previous knowledge, not her study that's currently going on, getting a better idea of how microglia function in the first place, because they found that some of them clear toxic proteins out of the body, out of the brain, like the amyloid plaques, which were already known to impact Alzheimer's. If the amyloid plaques build up, that can lead to Alzheimer's disease. And so they found that functioning in microglia, where if they're unable to clear as many amyloid plaques as they once were, then they'll build up and that can lead to it. And then they also know that particularly in developmental stages, microglia have a large impact in synaptic pruning. And that's great when you're a child and in the development phase and your brain is trying to figure out, okay, which synapses do we actually need? But the problem is one of the proteins which excites microglia to synaptic prune, or to prune the synapses, it's in high concentration in the developmental stages, and then it drops, and then the concentration goes back up in later stages of life, which can excite a lot of unnecessary neural pruning. And that's another reason why they think that Alzheimer's is such a problem, because you're losing synapses that you still need because of that elevated protein. Now, in the most recent research study that Dr. Stevens and her lab are doing, they're using the visual system in the mice brain. That's their biological model. And they're doing that because they have a really good understanding of how microglia work in the visual system, and also because they can activate one eye at a time or both eyes. They have a lot of control, and it's a smaller system, so they really understand it, and they're able to say, okay, we want to activate microglia in the left eye, let's go, and they can do it very precisely. So the experimental approaches and methods by which Dr. Stevens and her colleagues are going to do this new perturbation of high-risk gene experiment, it's probably going to involve, she doesn't get into the specifics, but based off of previous studies that she mentioned, I would think that it's going to involve a lot of protein injection in the mice brains in varying stages of development, and then after 24 hours of letting the protein settle and letting the microglia do whatever kind of function they're going to do depending on which high-risk genes were excited or inhibited, they're going to, you know, dissect the brain, and then they'll be able to see where and how the microglia were functioning in the different stages of development. Now, the implications of Dr. Stevens' current research are essentially astronomical, because they're trying to determine pathways which lead to Alzheimer's, and then figure out the microglia functioning in those pathways. And then once they know how it works, they can inhibit it and essentially stop Alzheimer's or even reverse the progression. And this is going to open doors for other researchers to say, okay, well, now that we understand this functioning of microglia, I'm going to use this to help my MS research, to help my depression research. And it's going to be really fascinating to see what kind of answers and questions this research leads to. And the implications of understanding microglia to such a broad extent will not only help prevent and restore people suffering from Alzheimer's. We know that the physical changes in the brain that microglia can lead to can cause depression and suicidal thoughts. And so the research that they're doing right now will lead to the questions of, okay, well, how do we use this information to stop depression? How do we use this information to stop suicide? And researchers will really have to focus on the methods Dr. Stevens has used in the past, and most likely in the future do more in vivo and non-invasive testing on brains to figure out, you know, whether they use cat brains, ferret brains, mice brains. Eventually, if they're able to get into humans, and they'll be able to keep answering those questions of, okay, well, now that we have this information, let's stop MS, let's stop depression. And so Dr. Stevens' presentation is very interesting, and I would definitely recommend it if you want a fun watch over the Christmas break. Thank you very much for having me in this class. I have really enjoyed it, and I've enjoyed your teaching, and I hope that you have a great break. Thank you.